name is the Golgi apparatus, and welcome to my crib. The Golgi apparatus is an organelle within the cell made up of flat sacs called cisternae. The Golgi apparatus is responsible for manufacturing, warehousing, packaging, and shipping cellular products within and outside the cell. Molecules are transported from the endoplasmic reticulum via transport vesicles, where they are then altered and packaged. Proteins from the Golgi apparatus are used in sending signals within the cell and repairing the cell membrane and performing functions outside the cell.
This is the smooth ER. It is a tubular structure connected to the rough ER. It's considered smooth ER because there are no ribosomes found on its surface. The smooth ER is responsible for synthesizing proteins and phospholipids, and detoxifying drugs and alcohol, and secreting hormones. Yeah, I think that's it. Shoot. Okay.
And this is the rough endoplasmic reticulum, or the rough ER. It is a continuous membrane system that, that forms a series of flattened sacs within the cytoplasm of a eukaryotic cell or animal cell, and is important in the biosynthesis, transport, and process of lipids and proteins. The ER usually constitutes for more than half of the membranous content of the cell and is continuous with the outer membrane of the nuclear envelope. The roughness of the ER is due to these ribosomes on the outside surface. These ribosomes are called the bound ribosomes.
Hey! This is the cytoskeleton. The cytoskeleton is the main organelle that helps the cell keep its structure as well as helping move other organelles within the cell. It is composed of microtubules, microfilaments, and intermediate filaments.
These are microtubules. The microtubules are the largest component of the cytoskeleton and are composed of alpha and beta tubin that form long hollow cylinders.
And this is the intermediate filament. This is the structure within the cytoskeleton that allows the cytoskeleton to keep its shape and rigidity, as well as be an anchor for other organelles in the cell.
These are microfilaments that are the smallest component of the cytoskeleton and form long helical shapes from G-actin protein.
This organelle is the central vacuole. It's responsible for storage and cell growth.
This is the nuclear envelope. It surrounds the nucleus by a dual membrane with multiple pores. These pores regulate the passage of macromolecules such as proteins and RNA, permit free passage of water ions, HEP, and other macromolecules. This membrane helps control the cell of information flow of the cell.
This is the nucleolus. It's a dense area of chromatin and chromosomes located in the nucleus. It is mainly a ribosomal structure, and you can see the granular forms of partially formed ribosomes. And the nucleolus' main function is to produce rRNA for producing ribosomes on the cell, and it makes these ribosomes, sips it through the pore complex in the nuclear envelope out into the rest of the cell.
This is a lysosome. It is found mainly in eukaryotic animal cells. Its main functions are to remove and digest excess food, worn-out organelles, and viruses and bacteria. It also recycles what can be reused inside of the cell. The proteins used to make it are made first in the Golgi apparatus, not the rough ER, which sends a transport vesicle to the Golgi apparatus, which makes the digestive enzymes found inside of the lysosome. After those are made, the Golgi apparatus pinches off a very small specific vesicle, which is the lysosome. The lysosome then floats inside the cytoplasm until it's needed.
This is the mitochondria. The mitochondria produces the energy a cell needs to move, to reproduce, or to produce fluids that it secretes. The parts of the mitochondria are the ribosomes, the DNA, the crista, and there's two membranes, because the mitochondria has two membranes just like the nucleus. The outer membrane of the mitochondria is smooth. The inner membrane includes folds called crista, which greatly increase the surface area. The increased surface area is where the oxygen and sugar meet to produce ATP, which is the cell's main energy source. The mitochondria is about the size of bacteria, but the shape will vary depending on the type of cell.
Okay, so if you notice, there's little red things floating all around in this room, and those are ribosomes. But in order to make it easier for you, I made a model that's bigger. This is a ribosome, and it's what it makes is proteins. A ribosome is made out of ribonucleic acid, or in other words, RNA. In the ribosome, the process in which proteins are made is called protein synthesis. This is done when the nucleus sends out information and it goes to the ribosome. In the ribosome are tRNA, and on one side of the tRNA are polypeptide chains, and on the other side are anticodons. The anticodons attach to the codons, and when they have successfully attached, they are sent out as proteins.
Peroxisomes are about the size of lysosomes and bud off the endoplasmic reticulum. Peroxisomes have a single membrane and contain a crystallized core. Enzymes and other proteins destined for the peroxisome contain a peroxisomal targeting signal. The signal then binds to a receptor molecule that brings proteins into the peroxisome and then returns for another load.
And this is the cell wall. The cell wall is made of cellulose fibers, proteins, and polysaccharides. It maintains and determines the shape of the cell, also the rate of growth, and it protects and waterproofs the cell. The cell, when it's new, has a thin, flexible cell wall, so it can grow easily. As it matures, the cell wall thickens and becomes a secondary cell wall. These are the plasmodesmatas. They are narrow channels that act as intercellular cytoplasmic bridges to facilitate the communication and transport of minerals through each cell. Straight hand out.
This is the centrosome. The centrosome is responsible for cell division and also produces microtubules. The centrioles are perpendicular, and they are responsible to replicate during cell division, and then the centrosome is made when the centrioles replicate, and the centrioles are also not found in plant cells. The microtubules are a long connected chain. They usually have two sets connecting the centrioles, and as you can see, there is always a break in the chain.
This is a chloroplast. It is the main site of photosynthesis and is only found in plant cells. There are about 200 chloroplasts per plant cell, and it consists of a double membrane and an inner fluid-like membrane called stroma. Inside the stroma, there are ribosomes and chloroplast DNA. It also has stacks of thylakoids called granum that produce starches and sugars for the plant to use.
This is the outer half of the plasma membrane. It protects the cell and allows entrance into the cell. It has proteins on its surface that act as receptors and can facilitate active transport. Outside the plasma membrane is the extracellular membrane, which is connected to the cell by fibronectin and is made of proteoglycan complexes. The extracellular membrane determines the properties of tissues.
And this is the inner membrane. It helps move waste across the cell and helps helps other molecules with their transport and serves as a border to the cell.